Now, that was very nice, and we tested these stimuli in large flies, that is Calliphora, as well as in Drosophila, and we saw the following. Here you see the responses of the blowfly and of Drosophila in response to on and off. We get positive responses, and we did not see any negative responses in response to these mixed stimuli. Here, even slightly positive responses in the blowfly, and a clean 0 in Drosophila. So this lets us exclude the 4-detector model and be sure that in the fly we're dealing only with 2 detectors, one for on, the other one for off sequences. So that's the answer to this question. Now, let me briefly wrap up and ask, what have we got so far? We started saying that we have the black box, that's our Reichert detector, and we asked the fruit fly to open it for us, and out came 2 other black boxes, right? So now we have, the workload has doubled, we have an on detector and we have an off detector. But that's, of course, sort of a negative view. Positive view is that we know several things. First of all, it seems like the splitting in on-off is like an evolutionary bestseller. We found that already in the vertebrate retina. It's been described for thermosensation just a year ago in Drosophila from Charles Zucker's lab, where you have on warm and cold receptors in the antenna of the fly, and if you increase temperature, you know, one goes up, the other one goes down. If you decrease the temperature, you know, it's the opposite, so they work in counterphase. And from the lab of Corey Barkman, that's also been described for chemosensation in C. elegans. But it also describes an easy way of multiplication. Here you see a grading moving to the right, and here you see the output signals of 2 adjacent photoreceptors that first go on and then off, depending on whether the on or the off edge has passed. And without any rectification and splitting, you know, your multiplier had to have this characteristic here that goes positive when both inputs go on and positive when both go off. But when you split the signals into separate multipliers, and in particular sign invert now the negative ones, then your multiplier only has to have a simple supralinearity, which is sort of which is easily done biophysically by any voltage-dependent ion channel. But most important for the next steps for us was that now we can use anatomy to guide these next steps. So different pathways have been anatomically described by other groups before, and so we simply look at who's postsynaptic to L1 and L2, and we find different neurons that then lead, for example, from the L1 through the T4 cells into the lobular plate, and from the L2 cells through the lobula via T5 cells into the lobular plate. So the obvious next question was, what happens if we block T4, T5? These were, so to say, long-standing candidates to provide the output of the motion detector onto the tangential cells. And we used a specific driver line that we got from Janelia Farm, Gerald Rubin's work, that expresses in both T4 and T5 cells. And Bettina Schnell, when she was still in the lab, you know, then did these experiments blocking with temperature-sensitive shibiri. Here you see the nice motion response of the control flies, and when you block T4, T5, it's a clean 0. And here you see the evaluation of a large number of flies, and these are different control flies. Here in black and in gray and light blue, you see the 0 response of the blocked flies. So we can now summarize what we found with respect to the cellular components of the elementary motion detector. First of all, we found that this computational structure is actually doubled in the fly optic lobe. We have one Reichert detector for the on and one for the off pathway. We have identified the input cells, L1 for the on pathway, L2 cells for the off pathway, and we have shown that the output of these multipliers then are represented by the T4 and T5 cells synapsing onto the lobular plate tangential cells. So we have not yet identified what neurons constitute the multipliers, what neurons do those interesting computations like temporal low-pass filtering, etc. So in order to sort of complete that picture between the L1 cells and the T4 cells, we are going to use the same strategy as we've used in the past. That is, we take specific driver lines to block these candidate neurons while using Shibiri, and then record at the same time from the lobular plate tangential cells. And with this, for example, we want to see whether our prediction is right, that T4, for example, really is an on-motion detector, and T5 then represents the output of the off-motion detector. But we can also use channel rhodopsin to activate optically all these different interneurons, again, while recording at the same time from the tangential cells. And when we combine that with this, pharmacology, this will allow us also to identify the transmitter systems. And finally, we use genetically encoded calcium indicators to record the activity of the different interneurons directly. And with all these different techniques, we hope to elucidate the cellular implementation of this Riker detector in the fly optic lobe that has been hidden there for so long in this jungle of this neuropil. Thank you for your attention.
Thank you very much, Axel. There is questions? I have a question. You block monopolar 1 and monopolar 2 type of cell, but what happened with the monopolar 3, monopolar 4, monopolar 5? Are they involved in motion detection?
So the problem is there's one of these monopolar cells called L4, and L4 is directly postsynaptic to L2. So any time we rescue or block L2, we also affect L4. So we cannot say anything specific about L4 at this time. We're now doing experiments with the driver line specific for L4 in order to say something about the participation of L4. However, L3 and L5 can be excluded because we did experiments where we blocked L1 and L2, and the motion response was completely gone. And we also did rescue experiments involving L1 and L2, and then we saw the full motion response. So L3 and L5 don't seem to be involved.
But were you able to block that neurons and see what happened, or
in behavior, you mean?
You haven't done.
Or with respect to other visual functions?
Yeah, or at the electrophysiological level to see if you still get some.
Which one do you mean?
M3,
L3.
M5.
No, we haven't done these experiments.
You haven't done.
So it is sort of an indirect evidence, what I
Okay.
summarized.